Assalamu alaikum everyone. I'm Sara Hamdani and welcome to this new video session where we will be studying about karyotype. Specifically, we will study human karyotype. So first of all, we will see what is karyotype. We can define karyotype as preparation of complete set of interface chromosomes in the particular organism cells. Okay. Chromosomes are then sorted and arranged on the basis of length, and then we can find what roles the particular chromosome and particular genes in that chromosomes are responsible for. Now we will see what are karyotype tests and how it occurs in its stepwise manner. Okay. What happens in karyotype tests? They take close look of chromosomes inside your cell and see if anything about them is unusual by comparing it to the normal set of chromosomes which is already present. They look for their size, shape and number. So basically it is visual depiction of chromosomes in the cells and prepared from cells just before they undergo cell division, that is during metaphase stage. Okay. Now, why typically only in metaphase stage? Because during metaphase stage, all the chromosomes are aligned at the center, so it is easy to distinguish and define them. If we don't have to search in whole cell, they are present in particular single line in the middle of the cell. This is characteristic of metaphase stage. So that's why metaphase chromosomes are preferred. Now let's see how is karyotype defined. Karyotype can be defined as accurate organization including matching and alignment of chromosomal content of any given cell type. In a karyotype, chromosomes are arranged and numbered according to size from largest to the smallest. What does it say? Organization including matching and alignment. Matching means they are matched from the one which is normal, means when you are going to karyotype your cells, your chromosomes, they will match it with the normal set, normal information which they have. And alignment means arranging the chromosomes according to their size from largest to smallest. So we can define this as organization of chromosomes including matching and alignment, arranging them from largest to smallest size. Okay. Moving forward, let's take a look of how karyotyping is done. We will see stepwise the procedure of karyotyping. So first, collection of blood sample. Blood sample is collected by using syringe which contains heparin. Heparin is an anticoagulant, means it protects your blood from coagulating. It will not clot when heparin is present. Okay, so injection is filled with heparin, blood is collected in that and it is put to centrifuge. By centrifugation it separates leukocytes. Okay, after centrifugation is done and leukocytes are separated, they are cultured in a medium phytohemagglutinin. Okay, phytohemagglutinin present in a medium in which leukocytes which are separated from by centrifugation from your blood sample are cultured. After culturing is done, there is step of arresting chromosome at metaphase stage. This is done at temperature of 37 degrees C by exposing it to particular chemical called colchicine. Colchicine is a mitosis inhibitor. It inhibits chromosomes at metaphase stage where we can, means cell division will be stopped, chromosomes will be arrested at that point. Okay, after we have arrested chromosomes at metaphase stage, we will prepare slides. After slide preparation, we will arrange, during preparation we will arrange chromosomes on the basis of their size, particularly from largest to the smallest. This is how karyotyping is done. This was all the detailed procedure of how karyotyping is done. Okay, I hope it is clear. Okay, moving forward, the next is the parameters which are used for characterizing the chromosomes in karyotyping. There are several parameters on which basis chromosomes are characterized in karyotyping. It includes shape of the chromosomes, length of the chromosomes, and centromeric index. What is centromeric index? It is a ratio of short arm to the length of total chromosomes. Okay, we know chromosomes has two arms, one is long arm and another one is short arm. So while calculating centromeric index, short arm is considered to the length of total chromosomes. This is how centromeric index is calculated. Now we know why, now that we have 23 pairs of chromosomes, individually 46 chromosomes. This was first classified by karyotyping in a conference at Denver in year 1916. Hence it is also known as Denver classification. Okay. According to this, there are 22 pairs of autosomes numbered from 1 to 22 according to their decreasing size, means on the basis of length. To 22 pairs of autosomes, there is one pair of sex chromosomes. There is a scientist named Patton. He divided the chromosomes into 7 groups, which were named as A, B, C, D, E, F, and G. Now we will see what these groups include, what are the characteristic features of particular chromosomes in particular group. Okay. So first, group A. Group A includes chromosome pairs 1, 2 and 3, and these are largest in size and are metacentric type of chromosomes. Next comes group B. Group B involves chromosome pairs 4 and 5, and these are smaller than group A chromosomes, and according to shape they are submetacentric chromosomes. Next comes group C. Group C includes chromosome pairs from 6 to 12, and it also includes X chromosome, which is a sex chromosome. Okay, they are all medium in length and submetacentric but smaller than group B. Okay, then there is group D, which includes medium sized acrocentric chromosomes number 13, 14 and 15 with one very small and one very large arm. They are all morphologically similar. Then there is group E chromosomes. Group E has two morphologically distinguishable categories. Chromosome number 16 is short and metacentric, whereas 17 and 18 are submetacentric chromosomes. Then there is group F, which includes two small metacentric chromosomes, chromosome number 19 and chromosome number 20, and these are distinguishable chromosomes. Then there is group G, which is last group, and this group includes very small acrocentric chromosomes number 21 and number 22, and it also includes another sex chromosome, which is Y chromosomes present in male. This is how seven groups are made for human chromosomes, and this is the result of karyotyping done at Denver classification. I hope it is cleared now. So let's conclude the topic. Thank you.